Hi guys, welcome. Hi Carol. Hi Chidima, how are you? How are we all doing? So what I thought I would do today, I'm going to do things slightly different. It's been a while since I've actually gone. Oh, hi Mum. How are you? Seeing me for live for the first time. My mom's with my sister, and so therefore she doesn't get to see what I normally do. This is where I just talk to myself until people actually pop in and say hi. So if you are watching me, then great. If you have questions, please ask away. But what I thought we'd do tonight is, it's been a while since I've actually done a walkthrough of a paper. So I thought what I could do is look at the 2023 paper. Oh no, Chidima, you have a cold. Oh no. Make sure you have honey, lemon, and hot water. Lots of rest. Ah, that's amazing. Your paper 2 is the best she's marked. That's really, really positive. That's what you want to be hearing. That's really good. Well done. That does make a big difference. I had some exciting news today. I've actually got a random interview tomorrow. How crazy is that? I haven't even told my. There we go. Mum and Carol, you can hear on here like everybody else. Yeah, I'm potentially interviewing to tutor students in Dubai. We'll see how that goes. So yeah, that will be some of my tutor slots potentially taken up for with students who are currently living in Dubai. So we'll see how that goes. Right. What I'm going to do is I am going to jump over and start actually going through a paper. So if you are if you are on my YouTube channel, then obviously you'll be able to see the paper. On here, I'll just go through what the questions are and we will work our way through it, unless you've got specific questions. Yeah, the Dubai, I mean, the international side of things will help with regards to filling up some of my daytime slots. Hello, me again. I was just asking, I picked biology, politics, and English. Could I still be a biology teacher? Absolutely. Because I know I can't do medicine or dentistry. So to be a teacher, you need to, depending on what type of teacher you want to be, you can go into the teaching profession first as an undergrad student, or you can go in as a postgrad student. It just depends which avenue you want to go down. I went the postgrad, and so therefore that was the route I took, and I loved it, and I love teaching. So yeah, it just really depends which way you want to go. I'm sorry that you can't do the medicine or dentistry, but oh, that's really horrible grainy picture on the YouTube channel, isn't it? Let me see if I can figure out what's going on there. Let me see what is the issue. I might need to do it a slightly different way. That's fine. Let me switch it around. We're going to do a bit of a switcheroo here to see if I can get this better. Is starting daily revision from February enough to move grades from a C, D to a B for the summer exam? Absolutely. Starting now is the best thing you can do. If you can start now, what's going to happen is you are therefore going to start getting what you need to progress. And so therefore I would say it's very important to start now. It's never too late. The more you can do, the more likely you are to be successful. The best advice I can give you for moving up the grade would be to take your time to figure out what you don't know first. Start revising the stuff you don't know, working through what is it that's missing from your answers that's going to help you moving forward. So is it that you're not given enough detail? Is it that you don't understand the question? What is it that's stopping you from being successful? So guys, I'm hoping this is a little bit better, but it does seem very grainy for some reason on YouTube, but I don't know why. So sorry if it's a bit grainy. Right, let's start with this question then. So what I would always advise whenever you are doing these types of questions is to try your best at figuring out what the question is asking, but by doing revision of the whole topic area. So many students will say to me, well, the answer is this, Christine, and I don't care if you know the right answer. That's not going to help you. What I care about is, why is the wrong answer the wrong answer? What is making the others not be correct? Because if you can't answer that question, that means you don't know your content. That means you don't know the information. Oh, I'm sorry, your mock didn't go so well. Thing is, the way to look at it, your mock is your indicator of where you're at right now. And so therefore, the key thing is use that as your guidance of what you need to do moving forward. Okay, so let's look at this then. Which description of biological molecules is correct? So we're looking at what's the right answer and therefore what are the wrong answers. So if we read through the question, so DNA and RNA are polymers of nucleotides. Well, that is correct. But if that is correct and that is our correct answer, I should be able to eliminate the wrong answers. And if I can't do that, then something is wrong. So hydrolysis of sucrose produces fructose and beta glucose. No, it doesn't. It doesn't produce the beta glucose, so therefore that is the wrong answer. Proteins are polymers of amino acids, excuse me, and are broken down in condensation reactions. No, they're not. They're broken down in a hydrolysis reaction. And it's really important as you're going through these practice papers that you are taking the time. So many students say to me, what can I do when I'm revising? And that would be one of the key things when you're using these multiple choice questions. You need to know how to eliminate correctly, because if you don't, you're not going to get the marks. So do make sure that you are taking the time with the revision to actually figure out what it is that makes that wrong, because then you're revising the whole topic area. You're not just revising the or getting the answer right or wrong, you are actually revising the whole area. Can you cover the topic which includes succession of plants? User, I actually have a whole video on that one, so if you haven't checked out my YouTube channel already, I actually have a video specific to that topic area in the playlist. So that is your ecosystems playlist that you want to look at, and there's one on succession there. Okay, carry on with this. Which statement about lipids is correct? So phospholipids are formed by esterification. Now it's interesting, they've actually removed esterification from the specification in 2023. I don't know why they decided to remove it, but they did. But phospholipids are formed by esterification of glycerol and 3 molecules of fatty acids. Well, it's not 3 because it's a phospholipid. I know that I have a phosphate head. I know that I have 2 fatty acid chains. Hi, Frankie. Thank you for joining me again. Thank you for coming and being part of my sessions. I do appreciate it. And if you have any questions as I go, do please ask away, and I will work through this. Right, let's move on to the next one. Phospholipids contain unsaturated fatty acids. Right, so first off, I would be saying, what does it mean by unsaturated fatty acids? So do you know what that means? So let's do a quick revision on that. If I have a fatty acid, I should know that my fatty acid has got a carboxylic acid group, and then I have, regardless however many carbons I bring in here, each of my carbons wants to have 4 bonds, and all those carbons like to have hydrogens attached to it. Now, if I'm looking at a unsaturated fatty acid, I know it's unsaturated because I will find a double bond somewhere. So if there is a double bond, therefore that means I've got less hydrogens. Now, for those of you who are not doing OCR but are joining me anyway and are wondering about how to tell whether it's a cis unsaturated fatty acid or a trans, if it is on the same level and produces what I like to call the C, then it is a cis unsaturated fatty acid. If you have got a double bond with the hydrogens and one's above and one's below, double bond with the carbons even, and it looks a bit like a cross, like a T, that is a trans unsaturated fatty acid. So phospholipids contain unsaturated fatty acids, can increase fluidity of the membrane. Now, if we have a double bond, that could potentially kink our fatty acid chains. So if I have a fatty acid chain which is going off in a different direction, oh my gosh, Frankie, you got your King's offer. That's amazing. Congratulations. That's really exciting. Oh, wow. Congratulations. I'm so proud of you. Well done. Make sure you definitely celebrate with a nice hot cup of tea or whatever it is you want to celebrate with. Okay, so coming back to the question, we have got the... Oh, I just realised I didn't actually finish the last one. Starch is a polymer of monosaccharides of maltose. Maltose is not a monosaccharide. Maltose is a disaccharide. See, even I didn't do it right. But always double check if you've answered it. Hi. Okay, so phospholipids contained in them will increase the fluidity. Yes, it can increase the fluidity because it can cause them not to pack together. So that one is true. So we think that one might be the right answer. But the only way we can know for sure is if we actually eliminate the wrong answer. So if you're just joining me, I am doing the 2023 paper 1 walkthrough on my channel. I'm going through how I would revise the topic area and how I would answer the questions. Okay, so triglycerides are good energy stores, although they release less energy than equal mass of polysaccharides. Well, that's absolutely rubbish because they release more because they don't have as much water associated with them. So we can get rid of that one. Triglycerides form the bilayer of the... Well, we can already get rid of that. We know it's not a triglyceride because a triglyceride is a glycerol molecule with 3 fatty acid chains. That's why it's called a 3 fatty acids attached to the glycerol. And we know that our bilayer is a phospholipid bilayer. So what I'm doing right now is I am... Oh, AB, oh, that's really sad that it was horrible. I have to say, doing the biology Olympiad can be really quite challenging. So well done for trying it. That's one of the things that you should take away from that you actually attempted something that was quite challenging. Now, the fact that it was horrible means that you probably worked harder at the questions, and that's a good thing. So I would use that as a positive for you in the fact that by having attempted it, even though you find it challenging, you've actually done something which is positive. So well done. It's a good form of revision. Right, question 3. Which statement about the structure of amino acids is not true? So which one is not true? So this is really important. So I would be going through this and I would be ticking what is correct and what is not correct, and then working out the answer, because this is something that seems to throw most students off. Whenever they throw in the word not, that is a big challenge. So amino acids contain an amino and a carboxyl group. So I know that my amino acid structure, and I would draw it out quickly here to remind myself what it looks like. And I therefore have an amine group on one side. So here's my amine group. Here is my carboxylic acid group. Here is my variable group. And therefore, an amino acid contains an amino and carboxyl group. So yes, it does have both of them. I don't like the fact that they've called it an amino group and they haven't called it an amine group. However, sometimes they do a bit of changing with the wording. My school has just finished module 2 for OCR A. Are we behind? It depends. It depends. I have a student that I tutor who their teachers started with module 3 and they didn't do module 2. So no, you're not behind. It really just depends which way around your teacher has taught the content. So I wouldn't worry too much about it. Okay, let's move on. Amino acids contain the elements hydrogen, carbon, and oxygen only. That's a really important one. Well, we know that's not true because we know that it doesn't only have that. They've missed out the nitrogen. So that makes me think that this one is the answer to this question, but let's just double check that we can eliminate the other ones. So each amino acid has a unique R group. Yes, because that is the variable region. And because it's the variable region, we know that there are 20 variations there. And so therefore, you can say, yes, each amino acid has a unique R group. The R group in amino acids can be polar and non-polar. Yes, it can. So therefore, we have eliminated all the other answers, leaving us with the only right answer, which is B. Okay, let's look at number 4 then. So, which of the options is a feature of collagen? So, straight away, I know that I'm going to make a little note to myself. So, this is collagen. Collagen, I know, is one of the ones that I have to know as my fibrous proteins. I know that my fibrous proteins are strong. I know that they're flexible. I know that they are going to be in the blood vessels and also to do with hair. So I'm just priming my brain, saying, what do I know? What sort of things do I want to add in here? So as you're going through, doing a little bit of blurting on the paper is actually a really good thing for you because it's going to help you to figure out where your knowledge is, to pull it forward and then go back and reread the question. A lot of students tend to look at questions like this, jump at reading the options, and then they misinterpret or they miss a marking point because there's a lot of similarities. So it allows alveoli to recoil. No, it doesn't. That's not collagen. That's elastin, which is another type of fibrous protein. So we know it's not A. B, it dissolves in water. Now that wasn't on my list. I know that it is insoluble. So I know it's made up of mainly non-polar amino acids. Now it wasn't in my list when I did my blurting. So because it wasn't in my list when I did my blurting, I would then highlight this because that is not something that came to my mind straight away. I was prompted when I read the next one, but it didn't come straight away. So this would be a technique that I would use when I was doing my revision. It's flexible. Yep, well, I said it was flexible, so that's potentially the right answer. But if that is the right answer, then I need to be able to eliminate the wrong answer, which is D, if C is in fact correct. It's the main protein found in here. No, it is not. The main protein found in here is keratin. So I know that that's incorrect, leaving me with the only right answer, which is the C in this case. Right, let's move on to the next one. So I'm doing it slightly different. I hope you're still able to see it all right, guys, on the channel. It's not too grainy. I don't know why it's been so grainy. It's not great, but anyway. Let's move on to the next one. Okay, it's not grainy for me, so that's really bizarre that it's grainy here. Anyway, let's carry on. Right, what have we got here? We have got increasing the pH of an enzyme solution from pH 7 to 8 causes the rate of the reaction to fall by 90%. When the pH of the solution returns to 7.4, the rate of the reaction returns to the original value. Okay, so this is straight away, again, if I was going to be doing this, I'm going to blur on the paper. What do I know? So this is talking about rate and it's talking about pH. It's saying increasing the pH from 7.4 to 8 causes the rate of the reaction to fall by 90%. When the pH of the solution returns, so when it goes, so going down, when it returns, the rate of reaction returns it to its original value. So straight away, I'm thinking to myself, okay, I've got pH. I know that enzymes are globular proteins. Globular proteins. Because I know that they're globular proteins, they are sensitive to pH because they will denature, but that denaturation is reversible. So because I know that denaturation is reversible, I know that if I go away from the optimum, it will decrease, and if I go back to the optimum, it will increase again. So I should note that if I were looking at this graph, I would expect it to do this. The optimum being the peak and the optimum being where the rate of reaction is the highest. With pH, if I go away from that optimum, I am therefore going to affect the rate of the reaction. Now, what do I mean? Well, again, in this case, because I'm looking at this question, what do I mean by pH? pH is the potential for hydrogen ions, or the potential of those hydroxide ions if we were looking at alkaline solution. In biology, we tend only really to look at the pH being to do with the hydrogen ions. So if I'm looking at those hydrogen ions, how many hydrogen ions are there? I need to understand that hydrogen ions are going to affect the tertiary structure of the enzyme. And by affecting the tertiary structure of the enzyme, they are going to affect the active site shape. And so therefore, that tells me that if I've got hydrogen ions affecting the tertiary structure, they are therefore going to interact with the bonds between the R groups. So let's have a look at what they've actually given me for the information. Hi, Adam. So what have they told me? They have told me, oh good, thank you, Mercy, it's still readable. Increasing the pH from 7.4 to 8 breaks hydrogen bonds, but returning the pH to 7.4 allows them to reform. Well, that kind of makes sense. I think that might be the right answer, but let's have a look. Increasing the pH from 7.4 to 8 changes the shape of the active site, and so the substrate binds more tightly. If the substrate binds more tightly, the rate of the reaction would increase and not fall. So it can't be B. Increasing the pH from 7.4 to 8 denatures the enzymes. Ooh, interesting. So it could potentially be that. So we'll come back to that one. Reducing the pH below 7.4 will reduce the rate of the reaction. Well, according to my graph, that could also be correct. So let's just double check what the question actually asked. And this is what tends to be where students miss it. I read the first sentence. I read the options. I didn't read the question. Which of the statements about this enzyme controlled reaction explains these observations? So which of these explains these observations? Well, what is being explained is when I increase the pH, the rate of the reaction is going to fall. That rate of reaction falling will then return to its original position. That would therefore mean it's all about the breaking of the hydrogen bonds and the reforming of the hydrogen bonds. So I would say that A is correct. C is not being explaining the observations that we've got. C would be explaining what has caused the first part of the observations. The fact that the rate of the reaction has fallen. So C only explains the first sentence. D is a way in which we're predicting what would happen. That isn't. So this would be a prediction of what we think is going to happen. That is not to do with these observations. So therefore, the correct answer would be A. Okay, let's go to the next set of questions. Annoyingly, we're having to adapt this. Right, okay, what have we got? Which of the options describes movement of water through a plant? Okay, I love this question. Well, not this question, but I love questions when it's about the movement of water through the plant. So, if it's talking about the movement of water through the plant, I need to be thinking about how does water move? So straight away, I know that water moves by osmosis. So that's the first thing I'm going to blurt on the paper. It's osmosis potentially. So we'll need to have a look and see if there's anything to do with osmosis. But I need to remember that water, if we're going through the roots, we're going to go through two different pathways. So we've got our symplast pathway and we have our apoplast pathway. So our symplast pathway, remember, is through our plasmodesmata. I'm running out of room here. And our apoplast pathway is through our cell wall and intercellular space. Hi! Space. So because we have our osmosis occurring, we have the symplast, we have the apoplast pathway, we then have transpiration, which is the transpiration stream, which is going to go up through the xylem, and we have the evaporation and diffusion, which is transpiration on its own. Okay, so all I've done is I've blurted right now onto the paper so that I'm looking at what have they told me, or in that first part, the movement. I've blurted what I know. Now I'm going to try and see if I can answer the question based on what I know. So the Casparian strip, okay, Casparian strip. What is the Casparian strip? Well, the Casparian strip, we should know, is a waterproof layer. Now, this is a waterproof layer which is found in the endodermis, and this is a way in which the plant is going to protect itself. So the Casparian strip forces water to enter the cytoplasm before it enters the xylem. Well, yes, that is true. But again, if that is the right answer, because I'm saying that's true, if that's the right answer, then B, C, and D must be wrong. So don't get fooled into going, oh yeah, I know the right answer, move on. That's not how it works. Multiple choice questions are really good if you know how to use them and if you use them to the best of your ability for revising. And that is, use them to eliminate the wrong answers. By eliminating the wrong answers, you're actually revising the whole topic area and assessing your knowledge of that topic. So if we look at B, how do we eliminate B? Water can move across the roots by the apoplast pathway because of the plasmodesmata between, no. Well, we know that's not correct because I've already said it's a symplast. Water is drawn up the xylem because of adhesion between water molecules. No, that should be cohesion between water molecules. So that C is wrong. Water moves by osmosis from a low water potential to a high water potential. No, it does not. They are the wrong way around. So these are questions that could be easy questions, which is a quick way of recapping, but by taking your time to go through a multiple choice question actually allows you to assess the whole content knowledge in your head. So do take your time. It would be one of the things I would advise. Right, let's have a look. Which of the statements about the heart is correct? Okay, we're talking about the heart. So I'm going to draw my little box. I've got my atria, my ventricles. What else do I need to know? Well, let's just go and have a look because they can go any way to do with this question when they're asking you about the heart. So make sure that you are not wasting too much because I could go and revise everything to do with heart, but let's see what they've actually asked me in this question. Contraction of the ventricle is known as ventricular systole. Okay, yep, that's true. Deoxygenated blood enters the heart via the pulmonary vein. No, it doesn't. It's oxygenated. The pressure in the walls is always greater. The pressure in the ventricles is always greater than in the atria because of the thicker ventricle walls. The pressure in the ventricles is always greater than in the atria because of the thicker. No, that's not true. It's not always greater in the atria because of the thicker ventricle walls, because when the pressure in the atria is greater, it opens the atrioventricular valves. So that's why that one is incorrect. The wall of the left ventricle is less thick because it only pumps. Well, no, it's not the left ventricle that's less thick. It's the right. I had to really stop and think on that one there. That was a one that made me, I had to reread it and double check because I was like, oh, hold on a minute. What are you actually saying to me? And again, sometimes the wording can seem so right that you confuse yourself. So it is important that you do take your time when reading the question. Right, let's look at this one. So the diagram shows the absorption spectra of different photosynthetic pigments. Right, we've got different photosynthetic pigments. They are looking at the absorption spectra. How much is the wavelength of light absorbed by those different photosynthetic pigments? Right, so we've got our wavelengths of light. We have our chlorophyll A, we've got our chlorophyll B, we've got our chlorophyll carotenoids. Now, I don't like graphs like this, and the reason I don't like graphs like this is because the lines all look the same. So I would always advise, to help me to actually be able to see the difference between them, I always highlight. Now, you're not allowed to highlight the actual answers that you have given, but you can highlight questions. So I would advise you, oh crumbs, that did not work. Let's try that one again. To highlight and have highlighters with you. So it's going down. Where are we going now? We're going a little blip up and down, and then we are going to go, let's go green, there we go. Okay, so we have got our carotenoids are our green, we have our chlorophyll B, which is our yellow, we have our chlorophyll A, which is our pink. Okay. In AQA, we don't learn what accessory pigments do, but I'm curious. So accessory pigments, you kind of do but don't. They're not explicit with the accessory pigments, but accessory pigments are pigments that can absorb different wavelengths of light. So these accessory pigments are going to absorb the wavelengths of light. They're able to then transfer the energy, the light energy that they've absorbed, transfer that energy on to the reaction center, which is where the electrons get donated. So whenever you've talked about excited electrons, you've talked about for AQA photoionization, and that photoionization is the excited electrons jumping up to a higher energy level. And so the other pigments are basically absorbing other wavelengths of light, and therefore that happens. What? This behavior is not. I love it. I just got a thing from TikTok saying that I'm not able to get any GIFs in TikTok because the feature is continuous and monotonous actions. Oh, that's hilarious. Very monotonous, obviously, not very good. Yeah, your teacher will have mentioned photosystem 1 and photosystem 2. So inside photosystem 1 and photosystem 2, you are going to have all these different accessory pigments that are passing the energy on to the reaction center. So yeah, it's hilarious. Very monotonous, very boring. Sorry guys, must be boring you tonight. Um, oh, anyway, back to this one. Right, let's have a look at what it's told us. So, which of the statements about the absorption spectra shown in the diagram is not correct? So again, let's just highlight that not correct because that is wanting us to find the inaccuracies. So in this case, we need to look at, right, absorption of light energy by accessory pigments such as chlorophyll B and carotenoids increases the efficiency of light harvesting. Well, absolutely, that does happen because what that means is that different wavelengths are going to be absorbed. Therefore, you're going to be able to use that energy for moving across the electron transport chain, pumping the hydrogens into the thylakoid space, allowing for the ATP to be synthesized. So that is a true statement. Right. Chlorophyll A absorbs. So let's look. Chlorophyll A is our pink one. So I'm just going to highlight that in pink so that I know we're looking at chlorophyll A. Chlorophyll A absorbs red light but not blue light, and chlorophyll B absorbs blue light but not red light. So this is where you then go, what's the red light? What's the blue light? I can't remember. Now, I was doing a lesson earlier on photosynthesis with my student. I said, oh, do you know what? Let's bring in a bit of your GCSE physics. And they were like, oh, really? Physics, GCSE physics. And I was like, well, okay, let's look at our electromagnetic spectrum. So you should know that visible light is part of the electromagnetic spectrum. And you should know that visible light can be separated out into the colours that we see in the rainbow. It's me, but I've changed to... Oh, okay. Yeah, it's what we see in our rainbow. So I always go Roy G. Biv. So I said to them, do you know Roy G. Biv? And they're like, no, I've never heard of that. Red shifts, so wavelength increases. So nice. So when you can bring in your GCSE knowledge of physics, it can help. Now, redshift means you were probably a triple student because you actually got taught about the redshift, whereas not everybody will have learned that. So remember, if you are combined science, that doesn't mean that you won't have the knowledge necessary. But it is important to go back and say, well, what is the wavelength? So if we're going wavelength, we're going peak to peak. I bring my wave down, or trough to trough. So this is the wavelength that is going to be between the distance between the waves, peak to peak or trough to trough. So whenever I do this, I think of it in a different way. So you've got your way of learning, red shift, so wavelength increases. I look at it as being, if I know my Roy G. Biv and I know after violet, I have my ultraviolet, and I know ultraviolet has got lots of energy, so therefore the wavelength is really small, that tells me I can do it the other way around. So therefore I would expect the top end to be towards the red and the bottom end here to be towards my violet. So Roy G. Biv, but back to front when you're looking at wavelength. So the shorter the wavelength is towards the violet end, the greater the wavelength is towards the red end. So it would be Roy G. Biv, and therefore I then also have that my green is about here, about 550. So that's how I remember it. But if you remember it, red shifts or wavelength increases, that helps you. That's what works. It's not one size fits all, and it's not learning one way works for each of you. It's about finding what does work for you and using that to support your learning and to help you learn in a faster way. Right, back to the question then. So if I know that, then let's just double check. Chlorophyll A absorbs red light. Well, chlorophyll A absorbs red light, but not blue light. Hold on, but there's a peak down. But not blue light. Chlorophyll B absorbs blue light, but not red light. Well, they both got two peaks, so that's not true. So therefore, that isn't going to be true. That's the best thing to do, is to use past papers. Past papers are great. The more you practice the past papers, the more you expose yourself to the questions, the more you start to see similarities. The people who are writing these exam papers are not going to reinvent the wheel. They are going to just adapt and change the question slightly, so that, I mean, they could give you this exact same graph, and they have in a number of exams, this exact same graph, and just ask a different question, but based on the same stuff. So if you can interpret it, that is going to help you. But if we're right, if B is the incorrect answer to the process of absorption spectrum, correct answer for this question, then I should be able to say that C and D are correct. I should be able to take the whole statement. So let's just double check that. Okay, so chlorophyll B and carotenoids are described as accessory pigments because they are not involved directly in the light-dependent reaction of photosynthesis. So they are accessory pigments because they are not involved directly, because they don't actually donate their electrons. Chlorophyll A donates the electrons, and so therefore that is why they are called accessory pigments. That is true. The absorption spectra explains why plants appear green, but because they absorb red and blue wavelengths of light. So the absorption spectra explains why plants appear green, because they absorb red and blue wavelengths of light. Well, yes, they reflect it. So they appear green because they are reflecting the green wavelengths more, and therefore they're absorbing the red and blue. In autumn time, that statement's not actually true when you see the leaves being different colors. However, what is important, that this is about this absorption spectra graph, so this is a correct statement. Oh, and therefore we can eliminate all the others. I was wondering if you could clear up the types of microscopes we just know for this year's exam. So the microscopes you just know for this year's exam are the light microscope, the 2 electron microscopes, the transmission electron microscope, and the scanning electron microscope. So you need to know the resolution differences, and what I mean by that is you don't need to know the numbers. You need to know that the transmission has the highest resolution and the highest magnification in comparison to the scanning and the light microscope. And you need to know whether it's 2D or 3D. So 2D is the transmission, 3D is the scanning. They've removed the laser confocal microscope. Yeah, I know, it's so cool. The different images, I used to do a starter lesson whenever I was doing microscopes with my students, where I would do them a little quiz and I would show them a picture and I'd get them to guess what the picture actually was. And it was using scanning electron micrographs, and it was awesome. It was things like a record, a salt, it was a flint, it was the leg of an insect. It was just super cool, all the things. And they were just like, what is that? Oh, that's horrible. Oh, and then they'd be like, oh, that's so cool. So yeah, it was really fun. It used to be one of my starter activities. It's funny to think that my life is so different now because I do all this online. It's not like I do as much as I would do in the classroom when I was doing it. Right, let's go. Next question. Let's see how we do on this one. Okay, I'm hoping these are still coming through clear enough for you guys on here. Okay, let's go. Right. The diagram shows the reactions that take place in a leaf cell in a flowering plant. So this one is important to note, that we're being told we're in a leaf cell, we've got a flowering plant, and we've been given some bits of information. Oh, you're going to freeze on me now, aren't you? Let's see if I can get you to, there we go. Oh, you're very welcome. I'm glad you're finding this a good use of time. It's really difficult to know how to revise and when to revise and at what level to revise to. And I think it's important that you do know that any revision that you do is going to help towards your exams, okay? But make sure the revision that you do is smart revision. It's not a case of you just sitting there mindlessly watching things. I mean, as I say this, as you watch me. But what I mean by that is actually engaging your brain to try and think through the process yourself. I could just tell you the right answers, but that doesn't help you. That's me consolidating my understanding. That's me strengthening my connections. You also doing that, you also trying it yourself, you practicing is a good use of time. If you're not sure, coming back and having a look at, for example, these videos, that will help you, and it's knowing what you don't know by using the diagrams given. Okay, so you've gone straight for, you think it's option C by using the diagrams given. So therefore, if it is option C, we must be able to eliminate A, B, and D. So if you think, oh crumbs, if you think C is correct, let's try that again. I must be able to eliminate B and A and D. So straight away, the first thing I can do is eliminate B and D if I can get rid of 1. So that's straight away. Oh, hold on. You're now panicking. You're now panicking because I said this. So this is why sometimes it's important to slowly work your way through and eliminate the wrong answers. So what have we been given? We've been given disaccharides and polysaccharides can go to glucose. Glucose can go to disaccharides and polysaccharides because we have got an arrow that moves in both directions. That's right. So we can have a condensation reaction, we can have hydrolysis, and that works that one out. We have got glucose becoming hexose phosphate, hexose then becoming hexose biphosphate, hexose biphosphate then becoming triose phosphate. Okay, we've then got our ribulose biphosphate. So over here, I've got my triose. I'm going to follow this one. I've got my triose going round to ribulose biphosphate and back again with carbon dioxide. So this is my Calvin cycle over here. Because it's my Calvin cycle, that therefore tells me that this is my light independent reaction. Now let's have a look at what we've got. So we had got glucose going down to triose phosphate, triose phosphate going to pyruvate. So this is showing us glycolysis. So again, I'm not actually answering the question yet. What I'm doing is is it not very clear? Hold on one second. Oh, yeah, I don't know why it's doing that. Let me zoom. Oh, no. Let me zoom in and see if that makes it any better. So what I'm talking about right now is we've got glycolysis happening here. So our glucose going to pyruvate is glycolysis. Wonder what's causing the blurriness. Not normally this bad. I'm not entirely sure why. I'll see if I can fix it for next time. Anyway, back to this. After glycolysis, just to move on, so if we go down now, we've got pyruvate to acetyl. So here is my link reaction. I've got my link reaction that's happening there. And then what I need to do is move on to my acetyl coenzyme coming in. So this is my Krebs cycle. And I've got the fact that different molecules can be utilized in different ways. So this is looking also, although we're talking about a leaf, we're also showing photosynthesis and respiration, but only a small part of it in the fact that it's a light independent reaction. So which of the following statements about triose phosphate are correct? So 6 turns of the Calvin cycle produces 6 molecules of triose phosphate. So if we have 6 turns of the Calvin cycle, so if we're having it go 6 times, are we going to produce 6 molecules of triose phosphate? No. We know that for every turn, we are going to produce the 1 triose phosphate that's going to go off, but we have, if we've done 6 turns, times that by 6, but we have 5 others that would also need to be replicated by 6. So that one is a really sneaky way in which they've worded that. So is statement 1 correct? 6 turns of the Calvin cycle produces 6 molecules of triose phosphate. No, because remember 5 for every 1 needs to go back to actually give us the RUBP, regeneration. So for every 1 that goes off, 5 must go on. So 6 turns of the Calvin cycle wouldn't produce 6 molecules of triose phosphate. To get 6 molecules of triose phosphate, we would actually need there to be more. So I would say that 1 is incorrect. But let's just double check that with the others. So triose phosphate from the Calvin cycle can be recycled. Yes, it can. Triose phosphate from the Calvin cycle can be used to make cellulose. Yes, it can, because we've been shown that it's going to be able to go up and across here, and we know that cellulose is a polysaccharide. We can see that it can go down to become glycerol and then lipids, and we can see that it can go down to become pyruvate, to become acetyl coenzyme A, to come all the way around here up to amino acids, and then up to become proteins. So that would make the correct answer be C, which is the 2 and 3 and not 1. And because we've removed 1, we've therefore eliminated it. So you were actually correct. However, you doubted yourself because you weren't sure if that would actually show you the correct answer or not, because you didn't do the elimination process. Right, let's look at this next question. For those of you who are just joining me, this is 2023's paper 1 that I'm looking at now. Because we are now on to the point where 24 and 25 are the ones I shouldn't really be showing you. I'm therefore free to use these ones to guide you. Okay, so what are we talking about? Well, we have got a neuron which is attached to an effector. Now, again, this is all about using the neurons, connecting it to effector. So straight away, the fact that I'm connecting to an effector means I am looking for a motor neuron. So I'm looking for a motor neuron because that is the one which is going to be connected to that neuron, that effector. And the reason it's connected to that effector is because it always goes a stimulus, which is detected by the receptor. The receptor then passes it on to the sensory neuron, which then passes it on to the relay neuron, which then passes it on to the motor neuron, which then passes it on to the effector. And the effector could either be a muscle or it could be a gland, and that effector will then carry out the response. So we always follow this same train of thought. So make sure when you are looking at this that you are noting it's giving you some hints in the question. So because it's given me some hints in the question, it means that I can then use that to guide my understanding. So what have we got? Well, whenever we look at a neuron, we should know that we have got our dendrites or our dendron. We have got our axon. We have got our cell body. So again, all I'm doing is I am blurting down words that are going to make the difference. I think there would be 2 of the line parts to connect the relay to the motor to the effector, but I don't know how the cell body. Okay, so it's a really interesting one. If I'm looking at my sensory neuron, so one of the things to note, when I'm looking at my sensory neuron, I would expect the cell body to be off to the side. So D is actually showing a sensory neuron. So that's the sensory neuron. Now the next thing I want to note is B does not have any dendrites. And because B doesn't have any dendrites, that tells me I'm not looking at any actual neuron that I would use as an example. A is showing the cell body in the middle, trying to confuse you, but we're not looking for a sensory neuron, so we know that A is not correct, which would leave me with the only right answer, which is C for this question, because C is showing me the dendrites leading to the cell body, the axon leading away, and the reason this is a motor and not a relay neuron is because that axon is longer. We would have a much shorter axon if we were looking at a relay neuron. Okay, let's go on. It's amazing. I've been going for over an hour and yet we've only got to question 11. And yet we've revised quite a lot of topic areas in a short amount of time. And so again, this is why I would say using these multiple choice questions is actually a really good use of your time because they will help you. So if you haven't learned the structures, it is important that you are looking at, and thank you so much for the likes, by the way, looking at all the topic areas. So that is animal response. Give me 1 second, I'll pull in a, here's one I prepared earlier for you, diagram. So this would be the one that I would always talk about. Hold on, I just need to move a few things. See if I can get this just a little bit. There we go. Right, here we go. So if you're looking at your different neurons, here's my cell, which is my sensory neuron. Here's my relay neuron. Here's my motor neuron. So what we're looking at is, where's the cell body? The cell body contains a nucleus. My relay neuron is shorter than my motor neuron. So that diagram that we had before, as well, looking at it, going back up here, you can see that there's lots and lots of axon terminals as well, which is another indicator. whereas this one's only got a very few numbers. So that's another way in which it can help you. Okay, so let's move on to the next question then. Question 11. So, here we go. Still on animal response. You could kind of see that they're grouping some questions together here when they're doing these questions. So we're still on animal and plant response. We're still on that same module 5.1.5, I think it is. I'm testing myself now, aren't I? But yeah, so that topic area. How come it is unmyelinated? Yes, that's exactly why. It's so short, it doesn't need the myelination. It doesn't need to have the action potentials occurring at the nodes of Ranvier because it's only got a very short distance for the action potential to go down the axon. And so therefore, it doesn't need to be myelinated. So yes, the relay neurons are the unmyelinated ones because they're really short and connected. Okay, we're probably going to finish on this question tonight, guys, because I can see the time is creeping up. But I will try, if I can, and do a video looking at some more walkthroughs. Maybe not live, because it depends if I have the time. But if I have the time, I will try and carry on this paper with the walkthroughs and put it on my channel for you so you can have a look. Okay, so in a phototropism investigation, oat seedlings, I got a fright there. Oat seedlings were eliminated from one side as shown by the arrow in the table. The table shows the results of 4 different experiments. So we've got experiment 1. What can we see? We can see that there is bending towards the light. Experiment 2. Shoot tip is covered with a light-proof cap and it didn't bend. So there has to be something up here which is going to detect the light. Right, we have got the light coming from the same direction. We've got an impermeable sheet inserted on the shaded side and it has grown upright. So therefore, whatever is causing the bending cannot diffuse through. And then the shoot tip has been removed and replaced on top with the permeable agar block. Therefore, whatever is released is able to diffuse. So diffuses. I say whatever. I know it's auxin because we're talking about tropisms. So what have we got as our co-results to choose from the options? Which option correctly explains the experimental results? Experiment 1 shows that auxin increases cell division on the shaded side. So if we're looking at, excuse me, the first one, experiment 1 shows auxin increases cell division on the shaded side. Experimental results, well, that's kind of true, but let's just double check. Experiments 1 and 2 show that photoreceptors are in the tip of the seedling. Yes, that is true because the photoreceptors are going to be the ones that are going to detect the light. And when I compare 1 and 2, and that's all about the comparison, when I do that comparison, that therefore means I can use that to show that the photoreceptors must be in the tip. So experiment 1 doesn't show auxin increases the cell division shaded side. It just shows that the light is causing the growth on the shaded side. So what 3 is preventing auxin from doing. So number 3 is preventing the auxin from diffusing through. So when you have an impermeable sheet, you are therefore stopping the auxin from being able to make its way through. So it's impermeable. The auxin cannot diffuse through. If it cannot diffuse through, we know that auxin causes cell elongation. We would therefore not have more growth on the shaded side. So what we expect is the cells on the shaded side are going to be longer. They're going to have elongated more than the ones on the side that's lit, and that's what causes the bending. So if the auxin can't get through, if it's impermeable, it's going to not be able to diffuse through. And so therefore, when I talk about it cannot diffuse or can diffuse, I'm talking about the auxin, the hormone. I went through and did the elimination process and I've ended up with B. Ooh, you're thinking it's B. Let's just double check. Experiments 1, 3, and 4 show that electrical signals, no. Stem elongation, no, because that would be gibberellin if it was stem elongation. Experiments 1, 3, and 4 show that soluble inhibitors, they're not inhibitors, of the elongation move down the illumination side. So absolutely right. The process of elimination leaves us with the only right answer. So yes, that means A is not correct. And the reason A is not correct, because experiment 1 shows that light increases cell division on the shaded side. Now I know that auxin diffuses to the shaded side and therefore will cause the bending, but that experiment does not prove that. And that's why it's really important that you're looking at the wording of the question and the wording of the answers, because that's the difference between you getting the mark or not. Also, what they do is they put responses here of what they won't accept. So if you were to describe phototropism and you were to say auxin increases cell division on the shaded side, that is not correct, because what auxin does is auxin increases cell elongation on the shaded side, and that is what they're looking for you to actually be able to do if you're aiming towards that A star. Right. I am going to stop there because it is almost time for my bed. And so therefore I need to have a rest because Wednesday is my busy night. So thank you to all of you who have joined me tonight. That's not what I wanted to do. Let's go away. And I thank you for joining me. I hope you enjoy the revision. Keep going with it. I know it feels difficult. I know it feels challenging, but keep going with it. It will make a big difference. And if you are back to join me next week, then I look forward to working with you. If you are doing mocks, if you're doing interviews, if you're doing any of the things that are going on right now, which it seems so busy for everybody, good luck. Breathe, accept the challenges, and try your best. That's all anyone can ever ask. But thank you all for joining me. Thank you for being a part of the community. It means I can do what I love. And have a good evening.